Hello dears, how are you? Hope all are fine and safe over your home. So, have you written the notes of the last chapter? Yes, many of you had sent me the notes and I'm very happy to see all of your completed notes. Very good children, keep it up. Okay, so you all know that we were discussing our second chapter, that is microorganisms, right? And we discussed the different types of microorganisms along with their examples, right? And I believe that everyone are writing and studying the examples, isn't it? I know you are very good children and you will be doing the as per the instruction what I have told. Okay. So today we will be dealing the next session of our topic. In the last class itself, I told you this chapter is meant for the whole microorganisms. What are their positive sides? What are their negative sides? So today we are dealing with the useful microorganisms. How microorganisms are useful to us. Okay, so we are going to see useful microorganisms. Can you name some useful microorganisms? Yes, somebody told yeast. Yeast is a microorganism. Which group yeast belongs to? Yes, they belongs to fungi. Yes, and also a large amount of bacteria are also coining as useful microorganisms. Okay, so first of all, we can see how bacteria is offering as the useful microorganisms. Okay, so I'm rubbing it off. Okay, and we can see it in detail how the organisms give or how they help us. Okay, so first of all, in making food. The main thing which helps in making food is bacteria. Bacteria help us in making food, especially curd. From where do we get curd? From milk, isn't it? So the formation of curd from milk, formation of curd from milk is offered by bacteria. And which type of bacteria? That is lactobacillus bacteria. Which bacteria helps in the formation of curd from milk? Lactobacillus bacteria. Okay, so we are going to see how they are formed. That is how the curd is formed from the milk. Milk is having a protein and that protein is known as casein. What is the protein present in milk? Casein. Milk is having a protein. It is this protein which clumps together, which coagulate together. Coagulate or clumps, both are same. Which coagulate together and they form curd. Understood? They form curd. Okay, we are going to see how this coagulation of casein is formed. Okay, casein coagulation is formed only when the milk is acidic. Only the milk is acidic, then only the coagulation of casein is promoted. Clear? Now, we are going to see how this milk become acidic. Did you see that we often see at our houses and all how our mother make curd from this milk? How do she make? By adding 1 spoon of curd into the milk. Curd is essential to make curd from the milk. Okay, why? Curd consists of this lactobacillus bacteria. Curd contains this lactobacillus bacteria. Okay, so when a spoonful of curd is added to a milk, warm milk, generally warm milk is used here. When 1 spoonful of curd is added to the milk which contain the lactobacillus bacteria, what happens? It coins it makes the milk to form acidic. The lactobacillus bacteria present in the curd will make this milk into acidic. Only when the milk turns acidic, then the coagulation of what? Casein happens. That results in the formation of curd. In your textbook, it is very well given. Okay. In your textbook, it is very well given. You have to study it clearly, write it in your notebook, put it in a box. Okay. Now, once again, I will tell the curd which contain the lactobacillus bacteria when added to the milk will turn the milk into acidic. That acidic milk will promote the coagulation of this casein and that results in the formation of curd. I hope it's clear to you. Okay, put it in a box. It's very important. It is often asked in the exam. Okay. Now, another thing. In your textbook, one do you know is being given. Did you see that? One do you know is being given. What it is saying? What is being told that? See, curd is added only to the warm milk. The milk should be neither cold nor too hot. It should be warm. Warm means when we dip our hand or finger in it, it should not hurt our finger. But it should be slightly hot. That is the lukewarm state. See, then only the bacillus bacteria could activate its function. That is, it turns the milk into acidic. Clear? Now, what is being happening? One more thing is that when we drink milk, we feel some sort of sweetness, right? So, some sort of, even though if we didn't add sugar, apart from that adding sugar, some sweetness is there in the milk, right? So, milk contains a sugar and that sugar is known as lactose. Lactose is the sugar present in the milk. Okay, when curd is added to the warm milk, it turns the milk into acidic, isn't it? So, when curd is added to the milk, it will turn the milk into lactic acid. This lactose with the sugar present in the milk will be turned into lactic acid. Clear? And that lactic acid in the milk will promote the formation of curd. Clear? That is also clearly given in the textbook. So, this curd contains lactobacillus. So, it is the role of lactobacillus which convert the lactose present in the, I am writing milk as lactose. The sugar present in the milk is lactose. Lactose will be converted to lactic acid. I am repeating it again and again because these are new to you. You have to write and study, and also they are quite important. I hope it's clear now. Okay. So shall we move to the next topic? Okay. These are the role of bacteria in making food. And that first of all we found bacteria helps in the formation of curd from the milk. Next is cheese and paneer. They are also milk products. You know that. How this cheese and paneer are formed? Cheese and paneer also formed by the action of lactobacillus bacteria and streptococcus bacteria. We have studied these streptococcus pneumonia and all in the last class, right? So you are familiarized with this term. So it is due to the role of this lactobacillus and streptococcus bacteria, cheese and paneer are being formed. Another one is the bacteria helping the tenderization of meat. What do you mean by tenderization of meat? What do you mean by tenderization of meat? This bacteria would act upon the tough meat and make it soft. Soft in the sense tender. Okay, that is known as tenderization of meat. Okay, then the next thing is yeast. As you told, yeast, which comes under the group of fungi, help a lot in the food industries, especially in bakeries, pastries, for the production of cake, bread, pastries and all, isn't it? So in that, what is happening? In this, yeast would be added into the flour mix, and along with that the flour will be mixed and it will form into a dough, and after making into a dough it will be kept in the warm place, and that will help in the lifting up of the dough. Okay, lifting up of the dough. Clear? Now actually what is happening there? How this dough is being uplifted. What is happening? During this, actually the yeast will be using the sugar. Along with the sugar, yeast is added into the flour mix. So yeast plus sugar will be added. So during the rising or lifting of the dough, the yeast will break down this sugar. And will result in the formation of carbon dioxide and alcohol. Clear? And this process of breaking down of sugar by means of yeast into carbon dioxide and alcohol is known as fermentation. What it is known as? Fermentation. So it is due to the presence of this carbon dioxide, the dough will be uplifted. When we touch it, the air will escape. Okay, that is being done in the industries of cakes and bakeries. And also yeast are used in our household life also, isn't it? Our mom and all would be using yeast for our idli batter, dosa batter in order to uplift. And also, even though we didn't use yeast and all, normally we use rice and urad dal for making our idli batter or dosa batter. So we are keeping that rice and dal for by mixing 8 hours. After making it batter, we are resting it for 8 hours or overnight, right? So that makes the production of yeast in them. That rice and urad dal, when kept for some time, yeast will be generated and that make the batter rise. Clear? And that's over. That's all about yeast. And another thing, we discuss bacteria, yeast. Now we are going to discuss algae. Algae. How algae are helping? Algae, some algae, seaweeds and chlorella. They are used as food substances because they are having a rich source of proteins. So they are taken as food asset. Clear? So that's all about in making food. How the microorganisms help in making food. Next category is in commercial use. Commercially, how they are useful. Okay. Commercially, commercially yeast and all are used in Which industries? In beer, wine, then acetic acid. What do you mean by acetic acid? Yes, our vinegar is an acetic acid, CH3COOH. Later in your chemistry period and all, you will study it in detail. Okay, so as a commercial product, yeast are used to produce beer, wine, acetic acid, alcohol and all. Now, how do they are produced? Say, yeast are grown on the natural sugars that are present on grains such as wheat, barley and all. They would be growing on the sugars of this grain. And again, fermentation happens, that we discussed, isn't it? The yeast will act upon the sugar of that grains and undergo fermentation to produce carbon dioxide and alcohol. Clear? We got alcohol. Clear? So that is the usefulness of yeast. Again, some bacteria are being used for the production of tea, coffee and other beverages. Okay, and some other are being used, some bacteria are being used for the production of lactic acid, citric acid and acetic acid and all. So all these things are given in your textbook itself very clearly. They are given as a point wise. Similarly, how you have written your notes for the last topic, that's how you have to write these two topics. So today we discuss making of food items and of commercial uses. That is the useful part of the microorganisms. And also you have to check upon the multiple choice question that is being given in page number 22. Okay, you do the MCQs in the textbook itself. No need to write that on the notebook. Okay, so I hope today's class is clear. Today, importantly, we discuss the equation of formation of curd from milk, the role of lactobacillus bacteria, and how the lactose, that is the milk sugar, is converted to lactic acid, that we discussed. I hope today's class is clear. It's quite easy, but many new things you have studied, so you have to go through it. Okay, thank you. Have a nice day.